right. Hello, everyone. Welcome on in. And look, the technology is working. Look at that, folks. Welcome into another edition of GSA on Twitch. My name is Balint, and this is our special guest today, Hassan Bukhair. Hassan is a member of the Early Career Research Program at GSA. He's actually a co-chair of the Multimedia Subcommittee. He's a postdoctoral researcher. He has his PhD, and he's going to talk to us today about his career journey, the science that gets him excited, and more. Welcome on in, Hassan. How are you doing today?
Thank you for the kind introduction, Balin. I'm doing great. How about you? How are you doing?
Doing good. It's I think both of us, Chad, were talking about before the stream of how we've been getting a lot of snow today, or snow warnings on and off, whether or not we were supposed to
That's
get
right. That's
snow.
right. Yeah. So nothing really happened, right?
Yeah, so
Great.
far, which is lucky. Lucky so
Yes.
far. So Hassan,
True.
before we
True.
really delve into what we get started, where can you tell us a little bit about where you are now, what your occupation is before? Then we're gonna turn back the clock and see when you got interested in the sciences.
Sure. Absolutely. So I'm a postdoctoral research fellow at Brigham Women's Hospital at Harvard Medical School, and I'm in the sixth year now of my postdocing, and I did my PhD in 2018. So what I basically do in my postdoc is to investigate how DNA damage repair or other nuclear mechanisms contribute to the aging process and also aging associated neurodegenerative disorders such as Alzheimer's disease and the related dementias. I use multi-systems approach. I use Drosophila genetics, I use mouse genetics, and also cell lines from humans and patients, right, to study how perturbations or you can say dysregulations of cellular signaling contributes to the disease. So it's like coming at these basic questions of disease pathology from different aspects, right, and connecting the dots to see what mechanism is very much conserved so it can be targeted for the downstream therapeutic targets.
Nice. And Hassan, just a little note for the audio. We're cutting in and out just a little bit there at the end of it.
Okay,
Now
all
that's
right.
again, I feel like if you point right forward, it doesn't cut out.
Okay. So I should be talking louder.
Yes, please.
Or... Oh yeah, absolutely, absolutely.
Thank you, Hassan. So
Thank you.
that's it sounds like a really complicated question that you're studying in your current lab, especially given how many organisms you work with. How do you decide on a day-to-day basis what organism you're going to work on that day? Or is it more that you set out to ask a particular question, and how do you end up, deciding with such a multidisciplinary approach what the best organism is to use?
Absolutely, Colin. Thanks for this question. This is really a great question. So I can give an example. For example, one of my projects, which has been completed and we have submitted a paper and it's in revision. And the project basically aimed to understand how different mutations in tau contribute to the disease. For those who do not know what tau is, so every cell has a cytoskeleton. It's like if you have a big building, it would have pillars in it and walls in it, right? They hold the building together. So a cell, which is a basic fundamental unit of life, has a cytoskeleton, right? And this cytoskeleton is maintained by certain proteins, right? And tau is one of these proteins. So why is tau interesting? Because like almost 100 years ago, when Alzheimer's disease was discovered or reported by Dr. Alois Alzheimer in Frankfurt, Germany, he saw there were aggregates in the brain of amyloid precursor protein, which is APP, and intracellular aggregates, which is tau protein. So back then, it was not known. This was only reported that there are aggregates in the brain, and these aggregates basically disrupt neuronal communications, and that's when neurons die. So just fast forwarding like almost like 60 or 70 years, in early 80s, it was known that there's a protein called tau, which is causing this aggregation, right? And in the late 90s, it was known that the tau gene has so many mutations And these mutations, when people have it, they get dementia on various uh, degrees of like how of worsening symptoms, right? So, for example, if you get a mutation which is um, very toxic, and then you might develop dementia very early on. So, when I joined in my postdoc lab, I was basically interested to understand the basic function of the gene itself. For instance, tau is conserved in Drosophila in mouse and humans. So I asked this question that since we have this wonderful CRISPR-Cas9 gene editing technology, why don't we edit Drosophila genome and mutate the same mutation that gives people dementia very early on? So that's what I did. I produced a tau mutant model for the first time, and we were very excited when this mutation happened. It took us 6 months to do it. But right after we did so many assays on the Drosophila brain, and we found there was an age-dependent increase in several pathological markers in the brain. Like it
So,
was really recapitulating.
just a quick question for you, Hassan. Why did it take 6 months? Like, what in the
Yes,
technology? Because when people say CRISPR-Cas9 and genetic engineering, it supposedly comes across as like this really easy thing that is
true.
done,
Yes,
but it is anything but easy, right? So what was
that's
the
right.
difficult?
Absolutely. So the major bottleneck is when you're designing a technology to target a specific gene, you first need to make sure that your guides are the machinery that is targeting the gene to cut it and to introduce mutations. It has to be very specific and very robust, right? For this, we have in vitro assays, right? So we test the efficiency of guides, and also we test on computer as well. They're computer programs. So this is the first step. Once you do it, and after that, you clone the plasmids. That takes you some time to produce and generate the plasmids. That would be essentially reagents that you would be using to produce your mutation. And in insects, it gets interesting because then you have to inject all these plasmids or reagents or the DNA cutting machinery one by one into the embryos of insects, right? And if you inject 100 embryos, and it is very unlikely that out of these 100, all of them would survive. So the success rate is almost 10%, right? So out of 100, 10 embryos would survive because they don't like foreign material to be injected in them. So we have these embryos, right? So we inject like 100 Drosophila embryos. These are insects, right? And they're very difficult to inject. So we inject them with a microscope, right? Because they're not visible to the naked eye. And then there's a very fine injection needle that precisely injects tiny, tiny amounts of the DNA into the embryos, right? So once this genetic engineering machinery, aka Cas9, the CRISPR machinery, and the guides are injected, right? And the next step is we let these embryos grow. And majority of these embryos, they die because they do not like things to be injected in them. If you inject 100, you would get like 10, 10%, 10 embryos. And then out of these 10, there would be very few who would be edited, right? Because you do not know if once you inject the Cas9 or the gene engineering machinery, it was successfully transported to the nucleus itself where the DNA is, right? So for that, you have to first of all inject hundreds of embryos, not only 100, right? And out of these, your success rate is pretty limited. Then comes the screening. So once these Drosophila who were injected and they were viable, they eclose, and you have to first cross them to another Drosophila strain because you want to preserve them as simple Drosophila genetics, right? This mutation is in one line, you cross them to a native line. So then if it was homozygous, it would be heterozygous, right? The mutation is preserved. And then there is another genetic trick in Drosophila research, which is the balancer to retain the mutation. I wouldn't talk about it in detail. If somebody's interested, I can explain what balancers are. So this whole process right now I explained, right, it is roughly 3 to 4 months.
Wow.
And after that, you have your founder flies, right? Founder flies, the ones that are the daughters of the generation that was injected. So these are the parent flies, right? And these founder flies then basically PCR, right? So you PCR them, right? Some of them to PCR is a technique that tells you if a mutation is there in the gene or not. And this PCR step is also cumbersome because you're basically amplifying the DNA region where your mutation of interest is, right? So it almost takes you a month, right? The whole DNA extraction, then PCR validations, and then for sequencing, and eventually you get your result. So the whole pipeline is from 4 to 6 months. But first, if you got lucky and you had so many flies injected, and then majority of these flies survive and they were viable, then you can shorten it to 3 to 4 months, right? And if you're unlucky, you have to start from the scratch again. There was no mutation, right? So it's basically um, a stroke of luck, right? And there's another caveat which people might not be familiar that in mammalian system, right, when you inject Cas9, Cas9 is an enzyme. It's a bacterial enzyme, and bacteria they love to grow at 37 Celsius, right? And Drosophila insects they love to grow at 25 Celsius, right? So this enzyme it does not function well at 25 Celsius. And that's why it's way more efficient in mammalian system and bacterial system where it was originally evolved in bacterial system, right? Because bacteria is working invaded all the time by these viruses, and then invented this Cas9 system, the other Cas systems to protect themselves from these invaders. Right? For them, it's perfect, right? But insects did not need it, right? Because they were not being invaded. They might have their own mechanisms to cope up with the foreign invaders like viruses. They have immune response and other systems, right? But not this. So that's another caveat. The enzyme does not work well, so the efficiency goes down. So it
So
takes a long.
what I'm getting, Hassan, is that the popular media that tells us that you can just genetically engineer something and it's just a throwaway sentence in an article is
Yes,
really a lot of work, right? And so and
indeed.
you're using fruit flies, which are an organism that people commonly study, and so you can imagine a situation where if you wanted to work on something different, like you wanted to look at an opossum, for example, and do the same thing, it'll take even longer. And so it's
Absolutely.
these
Yes.
aren't trivial things, even like in publications where it's like we made this mutant that actually is a lot of work that goes into it, and you were just describing to us like all the places where it could fail too in something that we
Indeed.
already know how it works.
Absolutely. Yes.
And so it's
True.
just really cool then, like how much work that you put into that, and then what the result was. Yeah, what was the of that paper that you were telling us about, like with the
Yes, yes.
multi-organism?
So your original question was like, how do I decide which model system is an ideal fit for the questions I'm asking, right? So
Yeah.
I started that tau protein or tau gene is very essential for Alzheimer's disease, and it forms these aggregates and there are mutations in tau. And we wanted to study what happens when we go back in evolution, right? Like 600 million years back, Drosophila were there and mammals did not evolve, but tau gene is still there, right? It has the residues which got mutated in mammalian genome or in humans, right? So I essentially mutated that uh, residue that gives early onset of dementia in humans, right? And we see striking differences in the brain. We see an age-dependent increase in several markers of neurodegeneration, such as DNA damage, deficits, speculation in the brain, like they're literally holes in the brain of these flies. And then we also see some modest behavioral anomalies, right? Memory. And we further did very sophisticated probing, which is called single cell RNA sequencing. So what you're basically doing in that, you take the whole brain of fly, you dissociate it. It would have neurons, it would have other cells, it would have glia, and you encapsulate each individual cell, and then you sequence the content of the cell, which is basically the RNA. It tells you how many genes are active, are upregulated, in a single cell in the Drosophila brain, right? So we did that. We compared it to the fly, which did not have a mutation, and our fly, which had a mutation. So we identified the genes and the networks and the pathways that were disrupted and dysregulated, right? So it gave us so many insights. But this Drosophila, for this question to answer if these mutations have an evolutionary role or the consequences of mutation in a very primitive organism could be recapitulated, right? So it gives a basic insights into the function of the gene itself, right? And now we have so many candidates, right? And these candidates essentially prove that some pathways are conserved, like people have shown in their findings in the mammalian system or in the human system, right? And we are seeing the same results in Drosophila, right? So these pathways are now a very lucrative target for therapeutics against tauopathies and dementia and other diseases, right? So for answering this kind of question, I think Drosophila is a perfect system, right?
So that's really cool. Is that you were able to mutate the residue.
That's right.
And like we said, that's why we use fruit flies as a model, right? But you could do the mutation and you
Absolutely.
could recapitulate a lot of the molecular phenotypes and like behaviors as well. And I'm just still blown away that you also had like the holes as well in the tissue. So like a lot of those similarities were coming through in
Indeed,
so many
indeed.
different
Absolutely.
ways, which is it's really remarkable to think that that's how well these things are conserved.
Yes, it's amazing, right? So there was a famous biologist who once said, if it is true for a bacteria, it is probably true for a human. I doubt it, but most of the pathways, right?
Yeah.
If it work in bacteria, right? And because biology builds upon complexity. So if things are complex and this complexity is there, so there would be another layer of like complexity. So it won't be resolved, right? So, and it is like uh, a big building, right? It has so many floors on it, right? And bacteria probably are on the first floor, right? And it's very primitive. But then the floors you go up and up, you take an elevator, right? And then the floors change, right? But they essentially copy the same model that was on the first floor, right? So that's how it is, right?
I like that analogy a lot of like within a building and then moving up and down in complexity, but the foundations are all still there. That's a good way to
Absolutely.
really nice way of
Yeah.
explaining it. Um,
Yeah.
so take us back, Hassan. Like this is amazing work, right? That you're just like you're telling us about that you've been working on. But what and when did you get interested in the sciences? Or when did you see yourself, as becoming like going down this path? Because it seems like you're very specialized, but also very diversified in the sciences. Like again,
That's true.
how did you get to where you are today? Like what was the beginnings, like pre undergrad? What was the excitement that
Yeah,
got you?
true. So as a child, like when I was probably in kindergarten, right, like 3 or 4 years old, I don't know if these memories are accurate, but I think they are. The very first like thing I remember was like me and my kids' friends, right? We had this epiphany that trees grow from ground, right? So, and we thought, okay, what if we bury some stones? Probably mountains. They also come from the ground, right? So you're burying pebbles and stones and ground, right? Watering them, and oh, there's nothing happening. There's no mountain emerging, right? That was the first failed experiment that I kind of did, right? And after that, like when I went to school, and I was generally interested in mathematics, physics, and like space science is a big craze. Like people want to be an astronaut, right? The kids want to be an astronaut, right? Uh, but something what really changed was that an uncle of mine, right? He had brought a computer, probably 1995, right, at home, and the first movie that was played on that computer was the Jurassic Park, right? So when I watched Jurassic Park as like 6 or 7 years old, right, and I see them explaining that they took this DNA from mosquito who had bit dinosaur, and then the blood was preserved in an amber, and they extracted it, and by doing gene engineering, they produced. This was fascinating, right? So I got interested in biology, and in the next 5 years or so, I was studying biology, and something else really happened. Um, was like when I was exposed to genetics, right? Mendelian genetics, right? We were studying about this Russian or Gregorian priest who was doing these pea experiments, and by a sheer coincidence, right, the gardener in the school, he had planted peas, right, in the garden, right? So we are studying the Mendelian genetics, and look, we are seeing the peas coming, the flowers and the varieties, right? This really invoked, right, my understanding about genetics. Wow, this is cool stuff. I should really study it. We had like a live demonstration of how he had figured out like 150 years earlier. So this is happening in early 2000s, right? So I definitely wanted to study biology, right? So I went to study bachelor's in biochemistry in Pakistan, and I did my bachelor's, and I was basically interested in understanding how genes interact and how these interactions basically contribute to the biology of a living being, right? All these tiny units in the DNA, right? They can make such a complex thing as an organism, right? So further, I go to Germany. I did my master's there, and then after that, I
So,
did my PhD there.
one moment, Hassan. Hold up. That was a big jump there. Um,
Okay,
so, bachelor's in biology, looking at again how the like really getting excited about how the genetics and the DNA systems work.
yeah,
How did you
yeah, it was
choose?
biochemistry. Yes,
How did you choose an undergrad, and then how did you
yeah,
choose a master's in Germany? Like where were you coming from in that transition?
yes,
Like
yes. So I did my like uh, intermediate, which is like A levels kind of training, right? So, and after that, I had so many options, right? I did physics, chemistry, and biology. So there was a very big prestigious school, right? Unfortunately, I got good marks, so I was open to so many fields. I applied in almost, I think, uh, 15 or 25 departments in that school, just to satisfy my self-esteem that how many can I be accepted in, right? And I was not accepted in biochemistry and biotechnology because it was the highest mark or like the department, but geology, zoology, botany, chemistry, and these departments I got in, right? So I was thinking, contemplating going to chemistry, right? But then one like last shot, I went to the Department of Biochemistry just to see if the merit list had changed. And because some students they did not join, they also apply to so many programs, right? And therefore, fortunately, vacant seats, and they pulled my name up in the merit list. I was so relieved because I really wanted to go biotechnology, biochemistry, study biochemistry, right? So yeah, on that day, I was happy I really got into that program, and it was really, really a fun ride for me because it was like a hobby, a game, right? You're learning so many projects and subjects, right? And so this was 4 years of my training. And after that, I got very interested in doing like advanced protein-protein interaction, a gene network interaction studies, like I said before.
so did you
For
have
that,
access to research as an undergraduate? Like were you in a lab?
yes, yes, I was in a lab, and my project in bachelor's was to understand uh, so the dolphin genome, right? So we have dolphins, which are freshwater dolphins, right? They're dolphins in Pacific, they're dolphins in Atlantic, right? Indian Ocean, but they're also dolphins in rivers, right? There's a dolphin in Pakistan, which is Indus River dolphin. There's a dolphin in India called Ganges River dolphin, and then there is a dolphin in China called Yangtze River dolphin. And then there's a dolphin in Brazil, which is the Amazon River dolphin, right? Or the Amazon pond, right? So these dolphins basically got trapped into these rivers from sea somehow, and they could not get out, right? And so this project was basically comparing by evolutionary genomics, right, which dolphins are very close to each other in their genome.
That is
So this is,
amazing, Hassan. Like I've
yeah.
never, I, I was not expecting you to tell me about dolphin genetics as the first project.
I was also really skipping it, right? Because it did not occur to me if this is of so relevance here, right? So we had tissue from dolphins and in the lab and an undergrad, like there were PhD students as well, right? So they were sequencing it, and I was learning like when you have sequences deposited at NCBI, which is a center where you can deposit genomes and you sequence them. You can sequence yourself, and then you take the sequences from NCBI and compare them by computer softwares or programs, right? And this is basically called phylogenetics, right? Where you study uh, the trajectories or the tree of life by looking at the genes and how the mutations occur. So the goal was to show that the Indus River dolphin is distinct from the Ganges River dolphin, right? And it did not go back to the basic rivalry of India and Pakistan because Indus is Pakistani dolphin and Ganges is Indian dolphin now. So they wanted to get more like funding to preserve this species because if it was raised to the level of a species, it would have more funding from WWF, right? So and they kind of succeeded in that, right? And I was just a minor cog in the big machinery there in the big lab, right? But I enjoyed it a lot, like the tissue coming from the dolphin, and then you're extracting uh, the DNA and then comparing, right? And so after that, I also studied growth hormones from cattle, right? This was another industrial
That's so diverse,
project.
so cool.
Yes. Yeah. So it was basically to figure out like if you sequence the pituitary hormones, right? So pituitary is a hormone which sits below the brain. So if you excavate the brain of any mammal, right, you would see a tiny pea like gland, right? That's called pituitary gland. It's a master gland in the body that secretes growth hormone and other hormones and does the hormonal regulation, right? So another project in the lab was to figure out like if we could identify uh, polymorphism, which is a fancy word of saying that if you can identify a version of a growth hormone that is basically more active. Yeah, growth hormone that is basically more active to give more meat production or lactation in cows, right? So that was also computational comparisons of genes, right? So these two projects I worked during my bachelor's, and then I was further interested to take this knowledge and build upon it and to study how genes and networks they connect to each other, and that's how I landed in Germany.
So how, I guess, how did you pick going to Germany? And like, is it just like going to the master's degree was next because it just was like in the
Yes.
European system? Like, why?
Yeah, so yeah, my brother had gone to the US for his master's in bioinformatics, right? And I knew that trajectory was tough. So back then you needed to do GRE, which is graduate record examination test, right? To do your master's or PhD. And also you needed lots of funding for doing master's in the US. It's so expensive, right? And you need to essentially sponsor yourself from a bank or a higher education institute and so yeah, I was not really looking for that option. So my basic thing was to go to Europe and get a master's or PhD there, right? Or probably do further science there. So which was kind of a lucrative option for me. And in Europe, Germany was very prime candidate because most I used to live in a student dorm in Punjab University, Lahore, right? And in that student dorm, many of the students from space science program, right? They were doing their bachelor's in space science, physics, right? They were essentially going to Germany, right? So all the time they were talking about in the cafes, yeah, Germany is great and this and that, right? So I started to dig further options, right, in Germany if I could uh, get master's programs acceptance there. And for that, I yeah started off to universities and then they sent me out applications. I applied in so many universities and got admission in few. Unfortunately, I also got a scholarship for my master's, right, in one of the universities. So then I thought I have to go now because they're not only offering me free program, they're also paying me to come, right? So that's all I got in Germany, and then I did my master's there in biochemistry.
Did you have to pick your area of interest beforehand, or was it that you were applying to particular schools and after that you could apply, like pick your area?
Yeah. So I was basically interested to do any master's in biochemistry, right, which goes deeper into uh, the protein studies and protein-protein interactions or how genes they interact with each other, right? So, and back then, like if I look back, like it was almost 10 years back, like maybe 13 years back, 2011, right? And in my naive mind, I was not really aware like how biology unfolds, right? So any fancy stuff that could really give me an understanding of the deeper mechanism, right? I was aiming at many programs, right? So I had no awareness at that time that I would be studying genetics deeper or going deeper into that. Okay, can you hear me okay now? Yeah, so like there was no particular preference for going for my master's in Germany, right? So I even applied to population genetics lab, right? Because I thought, okay, I have a knowledge of Drosophila, oh no, in dolphin genetics, right? So I can basically use this knowledge and go to a population genetics, right? But I had also knowledge from this growth hormone, right, research as well, right? So that is basically protein-protein interactions, right? How a receptor, a hormone binds to a receptor, and it opens up the whole network and cascade of things, right? So I casted a huge net while applying for masters, right? And it paid off, right? I got into programs, right? And then the one program actually offered me a scholarship. So if people they are looking forward to like jump to the next career stage from masters or even from bachelor's to masters or masters to PhD, right? I would advise that they should cast a wider net, right? Because right now it's the skill building. Um, yeah, I can take it over. No. So it's the skill building uh, step in your career, right? So particular questions in biology change a lot, right? When new technology comes and it opens up new avenues and uh, yeah,
Gotcha.
so basically, yeah, build up your career capital and then later you can decide like where you want to go.
So in moving, like we're gonna ask like what your research was focused on in your master's, but what was you say when you say the most difficult component of moving to, a different country like in Germany, like what was difficult and what would you have like what was a big takeaway that you learned from that experience?
Yeah. So the first difficult task for me was that it um, Pakistan, right? That when I was studying during my bachelor's, right, it was more tailored toward textbooks, right? That you are given a problem, right? And then there's a specific set of um, rules that you can apply to that problem, right? And for instance, if you have to study metabolism, right? And metabolism is mapped by these several pathways like TCA cycle and other cycles in the mitochondria electron transport chain, right? So people know about it, right? So they would ask you questions. Okay, what is TCA cycle? Detail some components, right? How it happens, right? So this is one of the textbook stuff, right? But when I went to Germany, they were doing science or education differently, uh, because so essentially like giving you very basic glimpse of a problem, and then you're free to address it however you want. You can go to the internet. You don't need to go to a textbook, right? If your answers make sense logically, right, and then it should be fine. So that's how it is. So it was kind of challenging for me in the very first semesters to go to that. abstract um, way of thinking, right? That you have to start from the scratch, right? You're not building on others' knowledge, right? But eventually it pays me off now, right? When I encounter new problems and I can really attack these problems from very new perspectives and try to solve them in very non-trivial ways.
It's almost like the difference, I guess, of you're given a puzzle with all the pieces in the bachelor's, but then in the master's, you're told to make a puzzle with no starting material almost, and you have to build
Absolutely,
like the
absolutely. You
beginning.
you nailed it, right? So you said it in like two sentences, which took me just 5 minutes to clever. Absolutely.
It's I think that's always, at least in my opinion, it's the one of the hardest components about learning to be a scientist is that way of thought and approaching these problems, because like you said, there's not really a textbook. It's just part of it's that luck, like you were talking about in the CRISPR, and then it's almost
That's
like
right.
an intuition that's kind of like learned into
Indeed.
you.
Absolutely. Yeah,
Yeah.
that's true.
So what was your research topic, Hassan? You've told us about dolphins and about
Yes.
pituitary glands and catalog. Where did you go next? What were you studying?
Yeah, so I started my master's in biochemistry in 2011 and 2012. So first, like few 2 semesters were basically you have to take lectures because you need to understand that you have done your bachelor's in biochemistry, but in master's in biochemistry, you need to develop advanced thinking, right? So doing coursework. But then we were also rotating in labs, right? And these labs were basically interested in different. So I went to a biophysics lab, I went to a simulation lab, right, where I was doing computer simulations, right, and then I went to a proteomics lab, right. And the story of going to a proteomics lab is very interesting. I went to a lecture. So in German, it's called Ringvorlesung, right? So Ringvorlesung is basically a rotating lecture in English. It's a literal translation. So what would happen is not one lecturer that would come and give you a lecture. There would be lecturers from different disciplines, right? They would come, give you a lecture, you attend it, right? And then, so for example, in one semester, there are 15 or 12 lecturers coming, and then you basically are now familiar with the mentality of 12 people, right? Instead in a trivial system, right? Then you have one lecturer or two lecturers, right, for the whole semester. You would be basically interacting how one big professor, right, sees science, or how he or she they convey science to you, right? So in that ring zone that I was attending, there was a young professor who came and he showed us um, spectacular microscopy videos in the classroom, right? So basically, there were tiny complexes, donut-like structures in the nucleus of a cell dancing, right? And he said these complexes are thought to be involved in Alzheimer's disease. They might be regulating uh, gene transcription or replication, other mechanisms, but we do not know as of now what they are. And maybe some of you could come in my lab and investigate what they are. And I was so fascinated when I saw that video. Like, I really like doing microscopy as well. So I send an email to this professor, and then he invites me to the lab, and then I interview with him. And that happened in 2012. And in 2018, fast forward 6 years, right, I was graduating as a PhD student from his lab, right? So I spent like
Wow. So
6 years of my life.
so is it one where the system was you started in a master's program and then eventually transitioned to a PhD in the same lab and
Yes,
same location in Germany, same institute?
true. So after I go to the lab of this professor and do my first and big practical, 3 months practical, and then after that do my master thesis there, right? And my master thesis had revealed some spectacular results, right? So, but it was not ready to be published, right? So it was like 2 or 3 figures of paper, and we needed more data. So the logical would be that I give over this project to somebody else, right? Or I continue this as my PhD process, right? So I decided to continue, and I was very much interested in it, right? And it was happening in 2014, and I do not know why. So how was it? So this was basically a complex that was reported by Thomas Tuchel, who's a famous neuroscientist at Stanford. And in 2014, he gets a Nobel Prize, right? And I'm working on it in master thesis. I was like, oh my God, this is the thing, right? It would get me probably a Nobel Prize or something, right? So, and because the guy who discovered it, right, he got it, but he did not get it for this. He got it for synaptic proteins, right? But it really motivated me to dig deeper into this question because this guy was very genius, like he's still there. So, and that's how I got into it. And yeah, I continued it as my PhD and got 2 papers out of it.
That's really cool. And I think it speaks to the importance of like the different styles of learning too. Like you were saying that there's the transition of different professors that were coming in for these lectures, and this guy comes in and they're just showing off this video of like the donuts, and it's like this is what I'm studying, and it's just like
Yes,
that was able to capture your imagination. You just kind of the the next 6 years you say are saying that just got really hooked on that.
absolutely.
What?
Indeed, indeed. So these complexes of donuts are essentially like your background in the screen. You see these circles, right? Imagine they're moving very fast, right? These are called nuclear condensates, if somebody is interested, right? They are now a widespread phenomenon in cancer research and neurodegenerative research, right? That nuclear proteins, they form these complexes, and these complexes move very rapidly. And then that's how the gene regulation, replication, transcription, and all these processes are done.
So are they supposed to be there, or is that only in a disease state?
Yeah, so biologically they're supposed to be there, right? Because that's how these big machineries of enzyme polymerases and transcription machineries, right? They move in big complexes because everything happens so fast, right? So we have so much going on in our genome. Right now, I'm talking to you, but I'm forming memories. I'm also processing your questions. So this is such a fast-paced information, right? So my neurons are really like producing so much, right? So the genes are being activated. So this machinery moves very fast. So what the idea was in my PhD thesis that this machinery is disrupted due to toxic events in a protein, right? That is APP. And maybe you remember I spoke about Alzheimer's disease before, that there are two pathological hallmarks, one coming from APP, another coming from tau, right? So APP is giving the aggregates, right? And this protein, I was investigating if it was controlling these condensates and these complexes, right? So this was my PhD project, and then my postdoc I jumped to tau protein.
That's really amazing. Just the whole story of being inspired by that class too is what I'm just like really like wow,
Yeah,
that's
that's crazy.
that's
Yeah.
really really cool. What was your most exciting finding, would you say, during this time?
Yeah. So during my PhD, basically the most exciting finding I could say was that we had so this APP, this protein I'm talking about, right? It is also fairly evolution conserved, right? And this protein, when it's cleaved, so it is regularly physiologically cleaved. It's supposed to be cleaved. Majority of the proteins in the cell, they are degraded by cleavage or other mechanisms, right? So it's a regular process because cells have to get rid of uh, old proteins from new proteins, right? That's the cycle of life. But some proteins, they're cleaved in a dysfunctional way, right? It's non-physiological. And this cleavage basically produces fragments of proteins that are non-recyclable, right, by the cell. So these non-recyclable fragments, right, they tend to accumulate within the cell or outside of the cell. Sometimes
Hmm.
the protein is secreted outside, right? And these are called aggregates, right? These aggregates, over the passage of time, right? Like this passage of time is decades of life, right? So when a person, so neurons essentially live forever. Like forever means like when a person is born, right? Because there will be neurogenesis happening until age 25. But
Sorry,
after
Hassan.
that,
Sorry, Hassan. These are what after the aggregates? You cut out again.
so these aggregates basically disrupt neuronal communication,
Okay.
right? And that's how neurons they degenerate, right? So and neurons basically when they kind of live forever. Forever means, right? So when a person is born and then neurogenesis is happening, neurons are forming, right? And until age about 25 in humans, right? Most of the neurogenesis has stopped. There's new evidence coming that hippocampus or other brain regions might be still rejuvenating, right? But it's not that much. So the neurons have to live whatever they have at their disposal, right? So if these aggregates are formed, right, at any age, right, and this is non-physiological, they're not being recycled, they're not being degraded, they would keep accumulating, right, until age 70, 80, and 90, right? And until there would be a threshold point, or there would be a point break where there would be onslaught of dementia, right? So essentially, like the neurons cannot further tolerate, right, these aggregates, right, and that's how they die and degenerate. So a key protein in Alzheimer's disease is underlying precursor protein that forms the extracellular aggregates, right? And intracellular aggregates are contributed by tau protein. And my most exciting finding in my PhD was to show that there was a phosphorylation event. For those who do not know what a phosphorylation is, so majority of the genes in the living organisms, they produce proteins. And these proteins do many central functions, right? Like they build a cell skeleton, they give us so many other central functions like enzymes, and they form our organs, right? So and these proteins are regulated by different kinds of events, right? So they're modified. They're electrically charged entities, mostly negative, right? That's how the interaction of one protein to another protein is regulated. So cells have evolved through the process of evolution to change the charge of a protein, right? And this changing in charge is basically adding a phosphate group and a acetyl group. And these are simply chemical compounds, right? And they're added to protein, right? The conformation of the protein changes. At first, a protein was interacting with B protein, right? If you add an additional negative charge by virtue of a phosphate group, and now it won't interact with B protein, it would now interact with C protein. This is a very clever way in the biology, right? Because humans have essentially about 20,000 genes and they produce almost 100,000 proteins, right? But protein-protein interactions, how proteins interact, right? Right? These can be essentially regulated by putting different charge groups in protein, and then they become like millions of possible interactions, right? That's how the whole physiology of complexity evolves, right? So my basic finding was that there was an event, a phosphorylation event of this APP protein that was formed in aggregate, and that phosphorylation event basically inhibited the generation of these aggregates in the brain. And
So the
also,
phosphorylation was the preferred outcome, so that you don't have the aggregates. Is that correct?
indeed. And so the protein was not cleaved. And also, these toxic complexes I was talking about, non-physiological nuclear complexes, their levels also were reduced in the brain when this phosphorylation event was there. Right? So this was kind of win-win situation, right? If you increase the phosphorylation of this protein, you might be able to rescue Alzheimer's disease.
Wow, that's really cool. That when
Yes.
so just a naive question for you, Hassan. How does one study the charge of a protein molecule and see this interaction? Like, that's not something you can see under a microscope, right? So, what did you need to do, just from a technical
Yeah,
perspective, to be able to ask this question?
absolutely. Yeah. So very first thing is like you can study by several biophysical properties, right? So biophysics is basically studying reaction kinetics. First example is reaction kinetics, right? You have an enzyme, and that enzyme basically catalyzes a substrate. Let's take the example of um, lactic acid, a lactate, right? So many people have this lactose intolerance, right? They did not evolve it, and humans are now evolving to have lactose tolerance, right? What is basically happening in that example is that when children they grow up, right, they lose this lactate dehydrogenase or maybe I'm misremembering, this enzyme that basically processes lactic acid, right? But some people retain it, right? So this is the very first like biology example, right, that you can say that this enzyme is functioning well, right? And it is there because the lactic acid, the substrate of that enzyme is well. So if you want to study enzyme kinetics or how an enzyme, which is essentially a protein, right, on its substrate, which could be a molecule like lactic acid or another protein, right? So what is the half-life? So for example, A protein, if it cleaves B protein, how many molecules of B would be there if you put A in it, right? If A is functional. If A is non-functional, if by virtue of phosphorylation or other mutations in A, right? You had modified A now, and then you insert non-functional or more functional version of A.
Hassan, you're cutting out again. Could you lean a little bit
Yeah,
more?
sorry. Can you hear me okay now?
Yeah, I think we just need you to stay at that point, that
Okay.
forward leaning.
Yeah. Okay. Perfect. Okay. So where did I cut off?
You were talking about like putting the two different types of proteins within
Indeed,
what?
indeed. So the question was, how do I study? How do we study as biologists, like how different proteins interact and what are the ways, right? The first
Yeah.
way is to do classical enzyme reaction, enzyme kinetics, right? So an enzyme that is functional, right? It would consume its substrate. If you put lactic acid and lactic dehydrogenase or another enzyme that basically cleaves or eats this substrate, right? So you know this is a functional enzyme, right? And then there are so many examples, right? So you can first do enzyme kinetics, right, on a protein. But what if the protein is not an enzyme, right? And it interacts, right? So for that, we do classical approaches in cell science. First of all, you can tackle it by Drosophila, oh, sorry, not Drosophila, by fluorescence microscopy. So here, what can happen, right? So so there are proteins that glow in UV light. These are called fluorescence proteins, right? They were first discovered in jellyfish by Roger Sheehan in 1965 or around those years, right? And what basically was discovered that uh, these proteins, once you put UV light onto them, right, they would glow, right? So biologists or biochemists or biophysicists, right, they figured out an ingenious way to use this fluorescence protein to study protein-protein interaction. So what is happening now? So you have this full fluorescence protein. You chop it into two halves. First half goes to protein A. The second half goes to protein B. If protein A and protein B, they come close together and you shine UV light on them, if they are interacting with each other, then you would get fluorescence. Do you get it?
Yeah.
Yeah. Yeah. If they're not interacting, if you have by any chance produced a non-functional version of protein A or B, and then you bring them together, or yeah, so and they're and still shine UV light, they won't work, right? So that's one way of doing it. Another way is to use antibodies. Antibodies are molecules which we all have, right? Mammals, and also it goes back to evolution. So antibody is a molecule, right, that basically binds to any surface receptor or any molecule that it has affinity for, right? So it is a very clever molecule engineered through the process of evolution to neutralize or to
Hassan, could you lean
interact.
forward again?
Yeah,
Thank
can
you.
you? Yes.
Antibody, yes.
So
Sorry.
yes, antibodies. So the second way would be to use antibodies, right, against proteins, right? So if you want to study if protein A interacts with protein B, you use an antibody against protein A and pull out protein A by a biochemical reaction that is called immunoprecipitation, right? So you're precipitating, you're enriching protein A. And now you use antibodies against protein B. if you see protein B on a Western blot, which is a fancy way of detecting proteins, then you have essentially shown that protein A and B interact because you are not enriching for protein B. You are only enriching for protein A, but protein B came along anyway. So that meant that both had interacted in a biological system, right? They're interaction partners, right? And that is the second way of doing it, right?
Nice. So
And
it's
there's so
not.
many ways I can go on.
No, that's so it's not necessarily that you're looking for the exact charge of the molecule and like almost doing
Yeah.
like magnetism between the two proteins, but rather it's these
Yes.
other kinds of ways to see how two are interacting, that you can
Absolutely.
visualize
Yeah.
it. Yeah.
So indeed. Yeah, these are the classical biophysical or biochemical ways of looking at interaction, but essentially you can do NMR studies, right? Where you really go deeper and do a spectroscopy on these molecules and compare once a protein is phosphorylated, right, or non-phosphorylated, or what, because phosphorylation is not stable. It can be, when you purify proteins, it can be gone. So what people do now, they mutate uh, the residue which is phosphorylated to a negatively charged amino acid, right? The proteins are made of amino acids, right? There are 20 of them which are essential amino acids, right? So essential are 11, but right, in biology we know there are 20
Sorry.
total
Essentially, what, Hassan?
essential amino acids, right? The
Amino
building
acids. Yeah.
blocks of amino acids, the protein, right? So, and serine, purine, right? These are the basic amino acids that are phosphorylated. What people do if they want to study how a negative charge, in particular serine, threonine, or tyrosine, that's another amino acid, right? That is also phosphorylated, affects the structure of the protein itself. They mutate this amino acid to aspartate or glutamate, which is another negatively charged amino acid that mimics the phosphorylation event that could basically, yeah, trigger the interaction. And then by simple biophysics, by looking at the structure of these proteins, you can predict how the structure of the protein change, right? When you substituted these amino acids with a negative charge.
Gotcha. So you cut out at the very end, Hassan. So I think, just so you know, I think your mic likes it if you talk head on and are not
Okay,
tilted to either side,
okay,
even
okay,
at a
yeah,
close
sure.
distance. But
Sounds
just
good.
to follow up with that, so you were looking at these protein interactions, and again focusing on Alzheimer's potential aggregates, right? So then, how did you choose the next step in your career path? So like, next you went to postdoc. Um, at your current position, I believe, at Brigham and Women's. So that's another jump to a different country.
Yeah,
And
true.
so
That's right.
lots of
Yeah.
questions there. Like, how did you, the choice again of jumping to a different country? Why the United States? Like, why, not staying in Germany? Just and how would you recommend to another student in that position? How would you navigate that? Like, how did you pick the ideal lab? And then not
Yes.
just in terms of where you were going, but also the science behind it.
Yeah, sure. So while I was nearing the end of my PhD in 2017, right, I was wrapping up my projects and also really looking forward to doing a postdoc somewhere, right? And I had identified some big labs, right, and also some cool labs that were doing the next logical step that I could do, right? So I was simply applying to these labs. So I interviewed in a few labs
Sorry, Hassan, could you
here.
turn a little bit?
Yes,
Yeah,
and
thank you.
can you hear me okay now?
That's
Yeah.
right. Yeah, dead center, just like that.
Yes. So I had identified a few labs, and these labs were basically the next logical step for me to go, right, after my PhD. And it was in the Christmas break of 2017. I was doing live cell imaging of these complexes I talked about, right? These fast-moving nuclear protein complexes, right? To see how their dynamics changes with different disease conditions, right? So I had identified that the nucleus. So these are nuclear complexes, right? And I was using fluorescence microscopy to track the cell live for almost 24 hours or overnight, right? So this is called live cell imaging. You put the cell on a microscope, and the cell is in an incubator that has supply of CO2. The cells can live in CO2, carbon dioxide, and you have temperature regulated at 37 Celsius. And then you image it every 5 minutes and so, and eventually you generate basic continuous scan, a compilation of these images that becomes a movie, right? So that this is called live cell imaging, right? In fluorescence microscopy. So I was studying these complexes and I noticed something weird. When these complexes are there in the disease condition, right? So the nucleus of a particular cell would be very round in shape, right? If you might have seen nucleus, I've seen images of nucleus, right? Would be very regular in shape. I was noticing there were invaginations in the nucleus, or the membrane of the nucleus was being pinched inwards, right? This was very perplexing for me. The next day, I report this finding to my PhD advisor. He got very excited, right? And we had never seen something like this ever. And we also did not know that somebody else had reported. So I thought, wow, this is a cool finding. So let's look it up on PubMed. PubMed, it has a collection of all the previously published articles, right? We go on PubMed and then we find one article that was from the lab of Melfini, which is here, my current postdoc lab, right?
Uh-huh.
And they had shown these nuclear invaginations happening because of tau mutations, right? I was studying, or my findings were from APP mutations. So both APP and tau are the contributor to neurodegeneration, right? And we were really amazed. Oh my God, this is like a eureka moment, right? Like both APP and tau are doing the same thing, deforming the nucleus. What happens when you deform the nucleus? You get accelerated aging. There's a disease called progeroid syndrome, progeria. These individuals have mutations in the nuclear proteins. And because of that, their nuclear envelope, right? This envelope is invaginated and it cannot function well, meaning that their gene transcription, replication, and all these processes, right? They cannot be uh, like they cannot be organized in a regular pace like a physiological cell would do. So that's why they age pretty fast. And since Alzheimer's disease is an aging disease, we thought it was logical, right? So I sent an email to Dr. Feeney, my current postdoc advisor, that I found this and this is exciting and your lab has shown it, right? I want to study it further. If you have opportunities in your lab, let me know. And yeah, so that's how I interviewed with her and ended up here at Brigham Women's Hospital, Harvard School. And now I'm in the fifth year. I've completed almost like one project and second project.
That is a really serendipitous
Yes.
finding, right? Like I love that story that you went from, a discovery in the lab to a PubMed search to a eureka
Yes.
moment, and then that's how you
Yes.
identified the next lab. Um,
That's true. That's true. Yeah. Initially, I was interviewing with big labs, right? But honestly, chances of getting a postdoc position at Harvard were tougher, right?
mhm.
And I think this also played a huge role in me being here because I had some data to back up what they had essentially shown here, right? So, yeah.
So when did like on your career trajectory of looking at like Alzheimer's and these protein interactions, when did you decide that this was gonna be like the big focus of your future, right? Because it's not only like in graduate school, it seems like you're kind of continuing on with a not the same question, but a similar style question in your postdoc too. Like what got you so excited about this question to continue on with it?
Yeah. So one thing which really got me excited was that, for example, during my PhD, this APP, this protein, right? So this protein or this intracellular domain of this protein was proposed to be an interaction hub more than 50 proteins. It's almost like 10 years ago now. Maybe there are more proteins than genes, right? Uh, so proteins are the gene products, right? So if we don't confuse it, right? So, and this was really mind-boggling for me that one protein is acting as a switch to control the nuclear processes and also like the cell surface processes, right? And how the neurons are firing their synapses, right? This is also controlling that. So, and wow, there's so much to learn from this one gene or protein itself, right? So I was really fascinated by these immense, intricate protein-protein interactions, right? So, and way down further, I came to investigate tau protein. And tau is essentially the building block of the cytoskeleton of the cell that holds the whole building of the cell together. And tau also does so many things, right? So, yeah. So it is like mystery within a mystery, right? And I thought, wow, this is cool. Like one mystery, when you solve it, it leads to 10 more mysteries, right? Like a chain reaction. So that thing really kept me going in that direction.
Especially like you said, it there being a hub, right? That it's doing so many things, and then they're kind of doing similar things too within the protein discovery. That's really it's really exciting.
Yeah, it is. It is. Absolutely.
So, what is, Hassan? I guess your current research question that you're studying in your postdoc, and then just as an aside as well, you were a postdoc when the big COVID-19 pandemic hit.
Yeah.
How did that affect your research?
Yes. So the first question, like, what am I studying in my current postdoc? So the current postdoc aims to answer that how nuclear processes such as DNA damage repair, right, they contribute to the pathogenesis of dementia, Alzheimer's disease, and other disorders, right? So for that, I use these multi-systems that I talked about earlier, right?
Sorry,
That's
Hassan, you're in and out again.
uh, for that, I use these multi-systems I talked about,
Mm-hmm.
right? So to understand how the nuclear processes or the modulation of nuclear processes, it contributes to the regulation of pathogenesis or the disease onset or the severity of disease, right? So basically, it is very little known. People know a lot about the cytosol or the how the cells interact on the genes. Mutations lead to several cellular anomalies, but it is not known how these nuclear complexes, the repair machineries, how their deficit contributes, right? It's a very complicated question, right? So this is what I'm studying in my postdoc. And about the COVID-19 pandemic, how it affected my career trajectory or my science, it essentially robbed almost like a year and a half of time. Yeah, from my postdocing experience. So I had all the reagents ready at the end of 2019 and to do the next experiments, right? And it was February 2020, right before the lockdown. There was this news of a mysterious virus spreading throughout the globe, and I was having beers with my friends, fellow postdocs, right? And they asked me this question: "What do you think of it?" I simply naively answered them, "It's just a flu, right? It would be over." Like a flu season, right? That comes, and then in summer this wouldn't be a problem. But I was very wrong. I did not know, right? And our labs were shut down for the next 6 months, and so we got back into the labs, and we had to really sacrifice our mice or animals, right? Because nobody could come take care of them, and only keep the minimal stocks that could be rejuvenated at a later time point. Back then, we did not know how that curve would flatten and when can we go back. So when things opened up, and it also took a lot of time to ramp up because in science you build up a momentum, right? And this momentum basically carries you forward. Like when you stop, take a break, 6 months, a year, right? And this break is not only coming from you, but the whole system shut down, right? To restart the whole system, right? And take the momentum, right? It's gonna take a bit of time. So it took some time, so papers got delayed, but fortunately, right, I could finish my project, right, which I was working on. It took longer time, right? But it is what it is, right? So we cannot change what is not in our control. We can just like try to do something about it.
I think that's an important thing to highlight of what you just said is that ramping up time as well as the slowdown time, especially given if you're working with mice or any kind of like even you were describing the genetic engineering of the flies that takes 6 months, right?
Absolutely, yes.
That's not gonna be just 6 months if you have to stop everything. You have to get the reagents back. You have to grow the flies back up. You have to,
Indeed, indeed, indeed. Yes,
and
yes.
so it just I think demonstrate how valuable time is of a commodity to everyone, including scientists, right? And it's
Absolutely, yes. So, for instance, we are highly dependent as scientists on several facilities, right? Like our reagents, they come from a facility. If they are shut down, then your project is further delayed, right? You're collaborating with a core service that they do tests for you, they do sequencing for you, they do produce reagents for you, right? And you do not have anything to produce. I mean, you could produce on your own, but it would take you some time to get all the reagents again, right? So that core is shut down because people have COVID or they're not coming in, right? So it really, really delays uh, the progress of science.
and if you're looking at mice too, I'm presumably you're talking about Alzheimer's and its effect on neurodegeneration and aging. If you had any aging animals, that could be another huge setback.
Indeed, indeed, absolutely, yes, it is. So fortunately for me, it was not a big problem because I had finished up my Drosophila work, right, in
Mm-hmm.
end of 2019, and the next step was to go to the mammals, right, and to repeat everything in mammals,
Oh,
ordering mouse, right, and right before I was about to place the order from Jackson Laboratory, this email comes from the dean that okay, we will be shutting down the whole Harvard system and so and so. So my PI, she advised me not to do anything now because we do not know, not to get animals now because we do not know how long would it be. So, and that really had to be delayed.
and then doing after though, like you said, was the difficulty of the ramp back up and the different like,
Absolutely.
yeah.
Yeah, sure.
So
Sure.
never easy, never easy. Uh,
Yes,
So in that time, like after COVID, you also joined the Early Career Leadership Program of the GSA.
that's right.
Could you
That's
tell
right.
us a little bit about what you do in the program and what you're excited about within the program in terms of
Absolutely.
projects?
Yeah. So I joined in the early career leadership program almost a year back, right? In the late 2020s, I sent in my application,
Sorry, Hassan, you're
and I
cutting out.
was I sent in my application. Can you hear me now?
Yep, now we can hear
Yeah. Okay. So I just need to talk louder, I suppose, because
you.
sometimes my tone goes down and then my mic doesn't pick up my sound. So my long-term goal is to establish an independent research program where I will understand how genes, their networks, or the regulation of their networks contributes to the process of aging and aging associated neurodegenerative disorders. Right? That's my vision, like to have my own lab and have my own people who work on the questions, what I'm interested in. Of course, they're interested as well. And so in my postdoc, I have gained um, good experience of that lab, other lab techniques, right? But then there's an aspect of leadership, right, that you can get uh, or you need to acquire basically from programs like early career leadership program. And these leadership skills are very essential, right, in formulating you or making you a leader in the field, right? So that's how I got interested into the joining the program, and fortunately I was accepted. And then I was basically interested to join the multimedia subcommittee because my vision is to make science very simple. Maybe you've noticed in our chat in the year, hour and a half, right? To break down science very, very simple so people can understand, right? Because I notice when I talk to these big researchers or my colleagues and peers, right? They simply talk about these techniques like people know about it, right? But that's not the case, right? Even you yourself are a PhD biologist, right? And there might be some terminologies, right, that might be alien to you, right? Like how the charge of the protein changes. I don't know, maybe you know, yeah, yeah. So what are the biophysical, yeah, properties? So my vision is to make science very simple for people. And I noticed, right, like because during COVID, right, there was so much misconception about things, and even before, uh, like in terms of nutrition and other studies, right, people really do not make science simple, and that causes a lot of confusion among public or the general audience, right? So that's how I got interested in multimedia subcommittee, where we are actively working on making science and genetics widely accessible and understandable to the public. For that, we have a podcast which is Genetics in Your Word. And in this podcast, we highlight the key research from the journals of Genetic Society of America, such as Genetics and G3. And we basically break down the contents of these papers so that they are digestible and understandable, not only for biologists, but also for people who are interested to learn more about genetics and biology itself.
And Hassan, why is that important to you? Or what do you think the outcome of that would be as if making science more accessible? What are the long-term broad benefits of that?
Absolutely. So if you look at my career trajectory, right? I'm like a kid from Pakistan, right? Was like if we go back in our I was growing like not growing like putting stones, hoping them to become mountains, right? And it was right tuning into National Geographic channels, right? Discovery channels. And my mother, right, was kind of mad at me that all the time I was watching the like a nerd kid, these Nat Geo documentaries and so, but she was happy that I was not playing video games, right? And it paid off because then it kept me interested in science, right? And I kept developing my career skills by going to prestigious institutions in Pakistan, Germany, and now in the US, right? So I envision when you make science accessible for a general public, right? So the next Einstein, right, or the next Stephen Hawking, right, might be around the corner, could really solve the problems, right? So you just need to inspire them. So that's my goal, to inspire the future generation of scientists, geneticists, or biologists, right? So we can solve the key problems in science and make this planet a better place.
I love that, Hassan. That's a that's a really really
Yeah,
beautiful idea behind it. And can you tell us a little bit about how you plan to do that, or how you're doing that with the podcast, and then anything else that, like for in the future that you're gonna be doing, like you think that you can incorporate into like your future lab or any element of your science that can take that sentiment of making science accessible and really apply it.
absolutely. So the first step about the podcast, right? Here I'm training myself from my peers in the subcommittee and also in the GSA ECLP program, engagement staff and the faculty there, like how I can like gain the skills of making multimedia subcommittee, the skills essential for multimedia technologies, right? And in the podcast, we have basically a mission to uh, make these articles, the research in these articles, very simple for the people, right? So it is understandable. So that is one thing that we are actively working upon, right? Because if you might have noticed from there's a common joke between a physicist and a biologist that they name their things very simple, like black hole. Big thing would be a black hole, right? And biologist would be deoxyribonucleic acid, DNA, right? So it is very hard, right, for people to engage into the biology subject. What a protein is, what is like gene, like how gene is different from protein. They're essentially the same. It's just the product of the gene, protein, right? And but yeah, so biology I think is kind of complicated, and I think it's not the fault of biologists or the geneticists themselves, right? It is because the sheer complexity of it, right? It is indeed a complex object, but it is the job of scientists, at least some of the scientists, to make it simple for the general audience, right? So that's what we're actively working upon in our subcommittee, that we want to make the genetics research highlighted in GSA journals simpler for the wider audience, right? And in terms of my future goals, so when I would have established ideally my own research program, right, I would actively struggle to make every discovery in my lab as simple as possible, right, to uh, the people, right? One of my scientific heroes is Richard Feynman, right? And he used to say, if you cannot explain it simply enough, right, then you do not understand it. And I really account myself to that principle, right? If I cannot simply explain something to anyone, right, then I do not understand. I need to go back, right, and make my understanding simpler, right? So in my lab or in my future program, right, I would actively struggle to communicate key findings of our lab on our website, make YouTube channels or any social media channel, Twitch streams, which would be active in the next few years, hopefully. So promote there and to engage public as much as possible, right? Because you never know, you could be inspiring the next Einstein or the next key scientist who could solve so many problems.
I love that answer, Hassan, of just like you when you were planting the stones and getting
Yes,
inspired by National Geographic or Discovery Channel, and then basically your research and your future being the conduit for hopefully
yes,
inspiring another generation of scientists. I think that's a
that would
really
be
important and really amazing thing to be aware of and striving towards on that work.
absolutely, absolutely true.
So I've got just a couple of questions left for you, and if anyone from chat has questions, feel free to ask. But Hassan, what is going to be your next step? You mentioned again like a potential research program that you want to lead, like a lab head. What are the big questions that you'd want to ask, and what organisms do you see yourself working with in the future, and is there an ideal location in the world that you would love to do this?
Yeah. So the first question about the research questions, right, that I want to answer. So what really takes me is the question: how different organisms on this planet cope up with the same stress differently. An example would be that you have these long-lived naked mole rats, right? They live up to 24 years almost, right? And their counterparts, the simple rats, they live up to 3 to 7 years, right? So their lifespan is significantly increased many folds compared to their not so distinct evolutionary cousins, right? So what changed that? Similarly, there is another paradox in biology which is called the Patty's paradox. So what does Patty's paradox say? So Patty's paradox is the bigger the organism gets, the lesser it has chances to get cancer. It is true for whales. It is true for elephants. It's true for giant sequoia trees, right? So why is it that such a big system, it should have more chances getting anomalies and breakdowns, right? But it's still like defying the principles of biology, if they are any, right? So these questions really check me. And in the next like 5 to 10 years or so, I will envision myself to use multi-system biology, like throughout the lens of evolution, to answer these questions by using like CRISPR engineering, right, to engineer new organisms or models for the diseases, and use the cutting edge omics technology. Like it's so fascinating how technologies have advanced, right? We can essentially sequence the entire content of the cell.
Sorry, Hassan. We
See,
can what with the cell?
so we can essentially sequence the entire content of the cell and see in the temporal resolution what was active in a cell, right? So, and this technology is not stopping, it is going way deeper and deeper, right? So you can not only sequence the RNA, now you can sequence proteins probably, and then you can combine it with even the modifications of the proteins I was talking about, and even on the DNA, right? So this is an avenue I would be chasing and pursuing, right? But of course, in the next few years, when I transition to independence, these questions might change as well. But the general idea is to go in aging research and biology, right? So prolong human lifespan and alleviate human suffering from neurodegenerative and aging-associated disorders. And the next question was about the setup or where I envision myself to be, right?
If it was no object, if you could pick anywhere in the world to run this, where would you pick?
Uh, I would probably be in Boston, like as city-wise, right?
Mm.
Yes. So Boston is really a dynamic city where you have very cool and prestigious institutes, right? And really, the mixture of so many people from different backgrounds, right? Biologists, engineers, data scientists, right? And they come together, right, and give you some great science, right? So if I get a chance, I would probably stay in Boston, right? But the other coast is also great, right? And there are also some great institutions in the Midwest. So everywhere, it's fine here in the North America and also probably in Europe, right? I lived there for 7 years as well. I could envision myself there as well. And if my research brings me to Asia, where I'm originally from, or Africa, right, I would happily go, right? Because there's so many things that can be investigated there as well.
Cool. So pretty much anywhere. Although there are some
Pretty much anywhere,
slight
right?
preference for
Yeah.
Boston, maybe.
Yes. And if Elon Musk gives us a chance to go to Mars, I will definitely take it as well. So,
Nice.
so I can go anywhere.
Cool. Well, Hassan, thank you so much. This has been a really, really awesome chat. I loved hearing about the origins of how you got into science with like the planting of the rock seeds, doing dolphin research, as well as the peptides on cattle, then moving into like really a trajectory in your master's and your postdoc work on protein interactions, aging, Alzheimer's, disease curing. It's a really, really amazing story, Hassan. Like,
Yes. Thank you so much for giving me this opportunity. Actually, now I reflect back, and yeah, it has been an awesome journey, right? For sure.
it really, like, I feel like this should be a movie. Like, just when you know, so you're
Yeah.
telling us about like I mean, just like the inspirations too of what got you into science, and that really that curiosity that drove you, and
Sure.
it's really cool to hear about that diversity, that like of your the backgrounds that you pulled in, and like how it's all feeding. And I hope one day you come back to dolphins as well. I think that would be a
Hopefully.
really like cool tie together in the end of the day of like, because I mean you also
Yes.
mentioned too relatives of the dolphins, like the blue whales, and that problem of like them a really large animal you'd expect to have more likelihood of disease states and cancer, but those giant
Absolutely. Yes.
animals don't have it. So maybe we can bring you back to
There's
the
a connect.
dolphins at the end of the
Thank you.
day.
Yeah, I will take a note of it and maybe write a grant on it. Yes. Thank you.
Is there anything that you would like to plug or say, or as we round out the stream today?
Yeah, I just wanted to like I don't know if it's an unsolicited advice, but I can give. I don't know. So for those who are really interested in answering big questions, right? I would simply advise just like be curious, right? Eventually, everything that you want to answer, right, you would get there. Be persistent, be curious, right? And it doesn't matter like how impactful the discovery is, as long as it is satisfying your inner curiosity, because based on that, you would build up the muscle or the knowledge or the strength, right, to go solve the next curiosity, right? So do not be dissatisfied or do not discourage yourself, right? That if you do not see results in the short term, right, it doesn't matter. Just keep going on, right? You would eventually go there if you're persistent.
I think that's beautiful. I think your story absolutely highlights that importance of being curious.
Yes.
And
Oh,
again, like the serendipity too. So many times, the when you're taking that course on genetics and the gardener at your school has been planting peas, like
yes.
that's
That's crazy.
a
Yes.
beautiful. Or even
Absolutely.
the genes, right, during your PhD work that you identify and you look on PubMed and you find the other person who's looked at
Yes.
something similar, and
Yes.
it's I think it's a
True.
highlight of the entire journey is just being excited and curious about scientific questions and maintaining that curiosity throughout an academic or non-academic career journey.
Absolutely. Absolutely.
Well, Hassan, thank you so very much again for your time today, and
Thank you so
for
much for having me, Balin.
the
Yes.
audience for watching us. If y'all have any last minute questions, feel free to put them into the chat. Otherwise, Hassan, at the end of each stream, we take the audience over to a new location to continue watching the stream. And I figured today, with the sense of curiosity that you were telling us about, maybe a good place would be to look at some baby owls and new owls that were born. I think that's always kind of reminds me of the National Geographic feel and
Oh yeah, absolutely.
look. Um, so if there's no other questions in the chat, we're gonna go ahead and fire up what is called the raid. We'll go over and take the audience to the Hoot House live stream to check out the baby owls. So we've got. Oh, hold on. Let me go ahead and put in the command. There we go. We've got 60 seconds before we take the audience on over to the new location. So for folks that are still here with us, make sure to tell those people watching the Owl Nest what we were up to today, chatting with Hassan. Hassan, thank you again for your time, for your insights, and just for being an advocate for science communication. I think it's extremely important, and you're doing big things. Thank you very much.
Thank you for having me, Balin.